Okay, can people on Zoom hear me okay right now? Maybe like a thumbs up or unmute and let me know if you can hear me.
Yeah, so we can hear you.
Thumbs up.
I hear you.
Okay, great. I'm gonna stay muted until See, I just said welcome. Welcome everyone to Isabella's Exit Seminar. So my name is Thea Whitman. I have been Isabella's advisor for the last couple of years, and I just wanted to give a little bit of background on what brought her here and where she's going next. So Isabella did her Bachelor's of Science in Molecular Environmental Biology with a minor in Forestry and Natural Resources. And then her next step, degree-wise, has been to come here to the Department of Soil Science for a master's degree. But it has not been such a linear path as there's been many steps along the way, which I tried to map out. So along the way, between then and here, Isabella has been a forestry technician with the Department of Natural Resources and Environmental Science at the University of Nevada, Reno in the Stanislav Tulum Experimental Forest. Then back to Berkeley to be a lab assistant in the Department of iBio at UC Berkeley. And then started doing some serious fire work as a fire effects crew member with the Grand Canyon National Park, subsequently a fuels technician with the Bureau of Land Management, and then back with fire effects at the Redwood National Park, which brought her actually also to Lawson Volcanic National Park, which is where the bulk of her research has been based. And then came to Madison, but even then took a diversion in the first summer between her first and second year to go work more in fire out at the Olympic National Park as a wildland firefighter. So you can probably sort of sense a theme here, a lot of applied work and a passion for understanding how these systems work, how fire affects ecosystems. These are a few photos from Isabella and crew up at Lawson last summer. And out collecting field data. And along the way, she hasn't just focused on the work she's presenting here. She also was the recipient of an Owen Allen Soil Environmental Microbiology small grant. Here's some of the lab work for that. And what I think is just like the coolest photo ever of our collaborator Keith doing a burn simulation here. And also along the way, it hasn't been all work, there's been a bit of play. So here's Isabella at the Bucky Badger soil augering competition. The team had among the best shirts, I would say, although I don't think there was a prize for that, but I feel like there should be in subsequent years. And then also here's Isabella at one of our labs, various outreach events. And just along the way, she's been such a delightful colleague to have in the lab. I think everyone who's worked with her would say it's been a real pleasure, and we're seriously going to miss her. She's actually, after this, heading out to Sequoia and Kings Canyon National Park to work in the fire ecology program. So directly applying some of the stuff that she's built up today, maybe not so much the microbial things, but I'm sure you'll find a way to work it in. And with that, yeah, welcome Isabella, and we're looking forward to hearing about your master's work.
Thank you, Thea.
Okay, so I'm going to switch my slides now. Okay, and then for people on Zoom, it's coming up just a second. Okay. Cool. Can everyone see the slides in Zoom and in person? Yes. All right. Thank you, Thea, for that lovely introduction. That was very generous. And thank you guys all for coming to my master's public seminar. As Thea mentioned, my name is Isabella Muscettola. I've done my master's here at UW-Madison in the Soil Science Department, and I'm going to talk today about that work, which is studying the effects of fire history and burn severity on soil bacterial communities in Andisols of a temperate coniferous forest. So I'm going to start by talking a little bit about what we know about how fires affect above ground and below ground biotic communities, and then give a bit of an introduction to the study area, which, as Thea has mentioned, is Lassen Volcanic National Park. So in most recent decades, there's been an increase in wildfire size and burn severity in the Western US, and specifically within dry mixed conifer forests
These figures are coming from a paper that looks at a forest within my study area. And what you can see is that in recent decades, the number of fires that are greater than 400 hectares, which is generally considered to be a large fire, has been increasing. And the area of fires that burn under high severity has also been increasing. And generally, these trends have been attributed to changes in climate in recent decades with warmer and drier summers and shorter winters with a lower snowpack. But it has also been attributed to changes in land management and a departure from the historic wildland fire regime of these forests previously. To give a bit of a visual of what it looks like to have a change in the fire regime in these forests, what I'm showing is a photo series of the same photo point taken over 100 years. And what you can see is that there has been a change in forest composition and structure in the past 100 years through the federal land management strategy that's also state by state of generally just overall fire suppression. Historically, these forests were dominated by large trees and generally were more fire resistant, like Jeffrey pine. And in the past 100 years, there's been a densification of these forests with increasingly smaller sizes of trees and higher tree densities overall. And I should mention also that historically these forests were adapted to high frequency and low to moderate severity fires. So a departure from that through fire suppression has led to these changes. And it has implications for ecosystem resilience. In 2012, this area burned in a wildfire and had overall high mortality and high burn severity. And these trees species are not adapted to this disturbance regime, so it has implications for whether or not these forests will be able to reseed themselves naturally. I visited this area. I didn't visit the exact same photo point, but to kind of give you an idea of what it looks like now, you can see that there's not a whole lot of tree regeneration of those conifer trees. So likely there are like the departure from these historic fire regimes is going to be leading to dramatic landscape level changes. Okay, so I've just kind of talked about how fire regimes affect above ground plant communities, and now I'm going to talk about what we know about how fires affect below ground biotic communities and abiotic factors in the soil. So what I'm showing here is a classic soil profile where you have an organic horizon that's dominated by plant litter inputs, and then mineral horizons that decrease in their carbon content as you go south. Why we care about soils in these ecosystems is soils are the largest store of terrestrial carbon, and microbial components within these soil communities are really important for nutrient cycling and forming symbioses with above ground plants to help like perform multiple ecosystem services. During a fire, the heat can vary both vertically in the soil profile and horizontally dependent on fuel loading. And soil heating, or I guess the intensity of heating, is generally described as fire intensity. I'm going to talk about fire severity, which is kind of a bit of, it incorporates fire intensity, but it is more so defined by the departure from post-fire and pre-fire conditions, the difference of those two. And it can be characterized by a lot of factors, including like the percentage of organic matter that's consumed, the overstory or like vegetative loss during the fire event. But just so you know, when I talk about heating intensity, it's directly the amount of energy that's released during the fire event. And burn severity is referring to the change or the magnitude of change that occurs in these forests. After a fire in the days to weeks, there's a loss of organic matter through the combustion at the soil surface. But there's also inputs of new forms of carbon to these soils. Pyrogenic organic matter is a specific form of carbon that's produced during fires, and it's defined by the incomplete combustion of organic matter and generally tends to be more aromatic than its non-pyrogenic forms of carbon, so it can be more stable in the soil. There can also be changes to soil pH, and I'm focusing in on pH within this presentation, in part because that's the soil property that I measured with my master's, but also because it's a really important predictor for bacterial community composition at a global and local scale. And so with fire, you can have ash deposition, and ash is an alkaline substance, so sometimes you can have an increase in soil pH with the occurrence of fire. And there's also microbial death, again, because you have heating that can exceed the thresholds that are allowable for microbial life, although that varies spatially again. And so at depth, you may still have significant pools of microbes that are still alive and not affected by the fire that can then recolonize higher soil profiles. Months following fire, you may start to have a regeneration of the understory vegetation and subsequently a reaccumulation of that organic horizon. There may also be a redistribution of the ash through wind and precipitation events. And there also are likely going to be changes to the soil microbial communities in their overall community composition, in part because fires can have lasting effects on multiple soil properties, including hydrophobicity, water infiltration, can change the moisture regimes of these soils, and can also have impacts on the total amount of carbon and nitrogen in these soils, which are all important for the habitat in which microbes exist. There also tends to be a decrease in soil respiration, which is reflected by this loss of microbial biomass. And this dampening of microbial respiration or soil respiration also increases with burn severity, where the greater the magnitude of change, the lower the overall respiration. Years following the fire event, you may see a recovery of the overstory vegetation and a potential recovery of soil pH. Sometimes it depends on the soil type, and it obviously also depends on the magnitude of change from the fire event itself, but generally pH can recover in soils within a year to up to 9 years post-fire. You also may see a slow recovery of bacterial community composition. Again, this is going to be dependent on the magnitude of change or the burn severity. But generally, bacterial communities are more resilient or are better able to return to their pre-fire state than soil fungal and vegetative communities. So they can return to their pre-fire state quicker. But a key lack of understanding in the literature is how recurrent fires and also the regime of a fire, so not only the occurrence, but also the severity of the fire itself, how those factors impact soil microbial communities and also other soil properties. Two previous studies that have looked at the frequency of fire in soil bacterial communities have found that it can be a significant predictor in differences in community composition, but those are just two studies in completely different ecosystems, so it's important to get a better understanding of how disturbance regimes are affecting microbial communities because they are so important in the soil environment in multiple ecosystem services. So that brings me to my central research question, which is, do previous fires and burn severity from a recent fire impact soil pH and bacterial community composition? Here, I'm essentially trying to understand whether there's a lasting legacy of past fire on soil bacterial communities and whether that legacy is distinguishable based off of differences in soil properties, specifically soil pH. The approach that I took to answer this question is by comparing soils with differing fire histories from Lassen Volcanic National Park 1 year post-fire. To give a bit of a background about what Lassen Volcanic National Park is, where it is, and what it looks like, it's in northeastern California at the southern extent of the Cascade Mountain Range. The elevation of the study area ranged between 1,600 to 2,000 meters, although the Lassen Peak is much higher in elevation, but this is just the study area that we were looking at. Also, again, the vegetation at Lassen can be quite diverse and expansive, but the areas that we're focusing in on here for sampling were mixed conifer forests with predominantly Jeffrey pine and white fir, although lesser components of incense cedar, white pine, and red fir, depending on the elevation. And then we also sampled from grasslands or shrublands where there was no overstory present. The study area is also characterized by a Mediterranean climate with warm, dry summers and cool, wet winters. To give you a bit of a frame of reference, this is not Wisconsin. Most of the precipitation that actually occurs during the winter in the form of snow. So these forests are quite dry during the summer, and part of that has helped to maintain a historic fire regime of quite frequent fires. Now talking specifically about the soils of Lassen Volcanic National Park, if you couldn't get it from the name, they are in fact volcanic soils. So a majority of the soils in this area are characterized as Andisols, which are soils from volcanic origin, and soils that have andic soil properties, meaning that they have high proportion of volcanic glass and short range order minerals. But there are also entosols and inceptisols within the park. I specifically focused my sampling on andisols, although there were some samples that I collected from entosols. But generally, all of these soils were volcanic in origin. They were also generally sandy to sandy loam in texture, and some of the samples were quite rocky, if you can tell by the pictures, like a high percentage of coarse rock fragments. Okay, now I'm going to get a little bit into the experimental design and the methods. So one of the reasons why we were so interested in studying or asking this question at Lassen Volcanic National Park is because of the history of overlapping fires within this region in the past 100 years. So seeing here in this map is a map of the park, and the red areas are places that had experienced fire between 1917 to 2020. And the darker red areas mean that there had been repeated fires in that area during that time period. Part of this is in fact due to the National Park Service's mandate to reintroduce prescribed fire within their parks, and that occurred in the late 1990s. So they started to get more interested in reincorporating a more natural fire regime in these areas. In 2021, though, the Dixie Fire burned through 2/3 of the park, and the Dixie Fire was, and actually still is, the largest single ignition wildfire in California recorded history. And so this is, again, kind of exemplary of that first slide that I was showing, where wildfires are becoming quite large and in generally higher severity. But another reason why we were so interested in studying this at Lassen is because of the Park Service's history of long-term pre-established plots that were established with the intent of monitoring the effects of fire within their prescribed fire units on overall ecosystem health, specifically looking at changes in forest composition and structure, understory vegetation composition, as well as fuel loading. And so those yellow points that I'm showing here are plots that were established and maintained by the National Park Service. And I paired those with the blue plots, which I'll talk a little bit more about how we created that experimental design later. But critically here, I just wanted to point out that we sampled from 3 discrete units of the park: the Butte Lake unit in the upper right corner, the whole unit, and then the Warner Valley unit in the southern part of the park. And all 3 of these units had different vegetation and soil types, but within a unit, we paired the site or the plots so that the soil and vegetation types were matched within a unit. Okay, so I'm talking about like more and less fire history. What does that actually mean? And it means different things for the different units. But critically here, it's that the more fire plots had experienced at least one fire that was not shared by the less fire plots. And so I said that means different things for the different units. So in Butte Lake, the difference in fire history is 2 previous fires that occurred in the more fire plots and didn't occur in the less fire plots. In Warner Valley, it is 1 past fire in the more fire plots that was not shared by the less fire plots. And in the whole unit, it's unique because the 2 most recent fires are actually shared by the 2 different fire histories, and the difference is 1 past fire in 2002 that occurred in the more fire plots. So Within a unit, we had these two different treatment types, and the more fire plots that I had mentioned had been pre-established, but the less fire plots we had established in 2022 when we went out and sampled. And the way that we decided where to sample was a randomized point generator in buffers of the same soil and vegetation type of the more fire plots. And these plots were at least 50 meters away from one another and at least 250 meters away from trails, roads, and bodies of water. At each plot, we had 3 quadrats where we measured various characteristics of the plot, including understory plant composition, the number of conifer seedlings that had established within the quadrat, and then a couple of burn severity metrics, which I'll get into later, as well as as well as taking a soil core, 1 core per plot. Or sorry, 1 core per quadrat. So a total of 3 cores in total at the plot level. The organic horizon for each of the cores we sampled in its entirety, and the mineral horizon we collected to a depth of 5 centimeters. We separated out these horizons and then measured soil pH and sequenced the 16S rRNA region of DNA, which is the barcode region for bacteria to identify them to a OTU level. And in total, we sampled 152 organic horizons and 185 mineral horizons. Okay, so as I'd mentioned, burn severity is a comparison of the post-fire versus pre-fire conditions, and it can be described by multiple metrics. And what we wanted to do here was describe burn severity based off of a couple of different metrics at different spatial scales, because many studies that look at the effects of burn severity on soil bacterial communities are only using the burn severity metric that's generated by satellite imagery. And we wanted to know if using different burn severity metrics would change the results that we would find. And so the first burn severity metric that we collected was a metric of the percent of combustion of the surface organic matter. And so this was collected at both the core level, which is the red circle, and the quadrat level, which is the white square. Unburned means that the quadrat or core didn't appear to have any form of combustion on the surface. But what's important to note here is that even though these samples were characterized as unburned, they still did occur within the Dixie Fire footprint, but they just didn't have any like visible charring or consumption of organic matter. Low was a partial consumption of organic matter, moderate was a more complete combustion of organic matter, and then severe was 100% combustion of organic matter, with also changes to the coloration of the mineral soil surface. At the quadrat level, we also measured the percent of exposed mineral soil, so essentially whether the quadrat had large proportions of bare mineral soil or rocks visible. And then at the plot level, within a 10th acre 10th acre area of our plot, we also measured canopy mortality, where unburned was less than 25% of the canopy had died during the Dixie Fire, low 25 to 50, moderate 50 to 75, and severe greater than 75% of the canopy had died in the Dixie Fire. And then again, coming back to the satellite imagery, this is, we used the monitoring trends in burn severity programs, thresholded DNBR, which is the delta normalized burn ratio. And this is a metric of the reflectance of land cover using near infrared and shortwave infrared bands of the electromagnetic spectrum. Okay, so getting into my research questions and hypotheses. The first thing we wanted to ask was whether previous fire occurrences and recent burn severity impact soil pH. And we hypothesized that high pH would be associated with increasing burn severity, and that high pH would also be associated with more fire occurrences. Looking at the soil bacterial communities as a whole, we wanted to know whether burn severity would impact the community composition. And we hypothesized that it would be it would be a significant predictor of bacterial community composition. Essentially the same question, but looking at a different variable. This time we wanted to know whether fire history had an impact on soil bacterial community composition. And we hypothesized that it would be a significant predictor, and it would be most strongly associated with differences in community composition when the number of shared fires is the lowest. So essentially the fire history, it had the most dissimilar fire history, or when fire history and burn severity were correlated. So an example of this would be that more frequent fire correlated with a lower burn severity of the recent fire. Next, we wanted to see whether bacterial taxa were differentially abundant with more history of fire and with increasing burn severity. So instead of looking at the community as a whole, we wanted to look at individual taxa of bacteria. And we hypothesized that bacterial taxa identified as differentially abundant with burn severity and fire history would be those that had been previously identified in past papers as responsive to fire. Okay, so getting into the results. The first thing that we looked at was whether burn severity and fire history were co-correlated. So essentially whether the occurrence of past fire had an impact on the contemporary burn severity of the Dixie Fire. And we found that these two variables were correlated within only one of our units at the Butte Lake unit, where the less fire history plots had an overall higher burn severity than the more fire history plots. And this kind of goes back to the central dogma of what we understand fire ecology within these forests as, is if you have more occurrences of fire, then you're going to be creating a more resilient forest that is going to have a lower burn severity with a recent fire. However, in the other two units, in Hole and Warner Valley, we didn't see a difference in burn severity based off of past fire occurrences. And this could be due to multiple factors. One, that the total number of unshared fires was only one in these two units, whereas in Butte Lake, there were two previous fires in the more fire plots that weren't shared in the less fire plots. The time between the fires also may be sufficient for there to be an accumulation of fuel that would essentially like negate any past fire occurrences and like decreasing that fuel. And in the Warner Valley unit in particular, personal communications that I've had with park staff and wildland firefighters that were on the Dixie Fire have led me to understand that this unit was the first area within the park that burned during the Dixie Fire, and it burned under quite severe fire weather conditions. And what I mean by fire weather conditions is like very low relative humidity, high winds. This area is also generally has a greater slope. And so all of these combined factors could be influencing the unit as a whole burning at higher severity. Okay, next, we're going to look at the effect of fire on soil pH. And so we found that soil pH does in fact increase with increasing burn severity. So this is in line with our hypothesis. And specifically, we found that the severe and moderate classes of burn severity had higher soil pH than the unburned and low classes of burn severity. And I'm showing this graph is both the organic and mineral horizons aggregated, but this trend is also true when you separate out by horizon and when you separate out by unit. So this is a trend that we're seeing across the data in total. One of the mechanisms that can be driving an increase in pH with increasing burn severity could be the quantity and the quality of ash that's deposited at higher burn severity. And ash I'm using to describe both the inorganic and organic constituents of organic matter that's transformed by fire. And so when you have greater heating intensity, the proportion of organic matter is lower. So essentially the inorganic fraction is higher, and the inorganic fraction is the portion that is in fact alkaline. So with greater heating intensity, which generally is like higher burn severity, you may have ash that's more alkaline that's deposited. And also potentially the quantity of ash is increasing with burn severity because you have a greater transformation of that organic horizon. Another interesting thing to note here is that, like I had mentioned, we saw this trend in the mineral horizon as well as the organic. And so this helps to support this idea that there may be some incorporation of ash into the soil matrix because we're seeing this trend in the mineral horizons as well. And this is interesting because in some studies, they found that 1 year post-fire, there's enough time that has gone by for there to be leaching of ash. And here we're showing that there may be some retention of that ash in the soil matrix. Okay, next, we wanted to know whether fire history had a significant effect on soil pH. And again, we hypothesized that the more fire plots would have higher soil pH than the less fire. And what we found was, in fact, that there was a significant effect, but in the opposite direction that we had hypothesized, where less fire plots had, on average, about a half a unit of pH increase compared to the more fire plots. And so what this is showing is that potentially there may be enough time during the fire-free interval for there to be a recovery of soil pH. And this could be driven by the leaching of ash through, again, wind and water erosion. And why we may be seeing greater soil pH in the less fire plots compared to the more, potentially could be that there may have been greater fuel loading in the less fire plots. And so a greater quantity of ash was produced in these areas. Okay, and so next I'm going to talk about the soil bacterial communities. And before I talk about that, I need to put a caveat here, which is that because of the nested experimental design and that we had 3 replicates per plot, we were concerned that including all 3 of those replicates in a model without accounting for the structure of the experimental design would lead to increased significant value of our variable of interest, essentially like that we would have pseudo-replication. However, the statistical test that we were using, this permutational multivariate analysis of variance, or PermaNova, didn't allow for the inclusion of random effects. So in order to not remove variance from our data by aggregating or averaging the community composition at a plot level, what we did was we randomly selected a core to include in a cohort at each plot in this PermaNova. And then we generated the p-value in r² of the variable of interest. And then we iterated that 500 times, randomly choosing a core per plot each time to include in a cohort, generated the p-value in r² values, and then created a summary statistics table, which reported the percentage of iterations with a significant p-value, and then also reported the mean r² and the standard deviation of the r² value for our variable of interest. So the first thing we wanted to know was which metric of burn severity is the best predictor of differences in community composition. And what we found were the smaller spatial scales of the quadrat and burn severity, or sorry, 4 and the quadrat burn severity of organic matter combustion were the best predictors of differences in bacterial community composition, with variance explained about 12%. And that was followed by canopy mortality, and then that thresholded DNBR, which is the satellite imagery metric of burn severity. The worst predictor was the percentage of exposed mineral soil. And this may not be super surprising in these forests because of the pre-fire condition potentially having like more spatially heterogeneous cover of organic matter. So a high proportion of exposed mineral soil post-fire doesn't necessarily mean that there was a high heating intensity. It could have just been that that area didn't have organic matter to begin with. So this is super interesting because not many studies look at how burn severity at different levels and at different spatial scales can be correlated with bacterial community composition. And it's perhaps not super surprising that the smaller spatial scale is a better predictor for differences in soil bacterial community composition, because that may best exemplify the habitat in which the microbes are actually living. So for future studies, potentially if you want a better understanding of how burn severity is affecting your soils, use a smaller spatial scale. Okay, next, we wanted to look at how burn severity affects bacterial community composition at all three of the units. And so what I'm showing you first is the Butte Lake unit, and I just want to walk you through how to read an ordination like this. So this is a principal component analysis, and each point represents a soil sample. The color of the point, it represents the burn severity. So a lighter color means a lower, and then a darker color means a higher burn severity. And then the shape of the point represents the horizon in which that sample was taken from. So square is organic and triangle is mineral. How close these points are to one another is representative of how similar those communities are to each other. So if the points are farther apart, the samples are more different than if they are closer together. Okay, so what we found was that burn severity was a significant predictor of bacterial community composition in the Butte Lake unit in 100% of the iterations of that permanova. And in those permanovas, it was quite high. The variance that was explained, 26 and 17% of the variance was explained by burn severity. And for bacterial communities, that is quite high. So it is a strong predictor of differences in community composition in the Butte Lake unit. However, in the Hole and Warner Valley units, we didn't see that it was consistently a significant predictor of community composition, with a majority, meaning greater than 50% of the permanova iterations returning a p-value greater than 0.05. There's a small caveat there, which is that a non-trivial percentage of those permanova iterations did return a p-value of less than 0.05, with about 42% of the mineral iterations in the whole unit and 36% of the mineral and organic iterations in the Warner Valley unit returning a p-value less than 0.05. So I'm saying an inconsistent significant predictor of bacterial community composition. And why are we seeing that it is a strong predictor in Butte Lake and not in the Hole and Warner Valley units? In the Hole and Warner Valley units, we are missing the lowest and highest burn severity classes. So in the Hole unit, there was one sample that was collected that was characterized as severe. And in the Warner Valley unit, there were no samples that were collected that were characterized as unburned. And this is more of a reflection of what actually exists out in the environment. It wasn't just like a strategic sampling. But potentially here, the lowest or the highest burn severity classes may in fact be driving that significant effect. Okay, so I'm showing the same ordination, but this time the points are colored based off of fire history. And so this is asking the question, does fire history impact soil bacterial community composition? And we found that, again, it was a significant predictor, but only in the Butte Lake unit. And again, it was quite high in the variance that was explained. However, after we controlled for burn severity by including it as a covariate in our models, the effect of fire history was no longer significant. And going back to our first finding that burn severity and fire history are co-correlated within this unit alone, it's difficult to disentangle those effects with our data, but it's not possible to say that the effect of fire history on contemporary burn severity is like, they may in fact just be entangled as an effect of past fires. And this is supported by a previous study that has looked at the Dixie Fire burn severity in its entirety and found that areas that had burned under low fire severity in previous fires burned under low fire severity in the Dixie Fire. So that's the one caveat there, that burn severity and fire history may in fact just be entwined in this unit. But as I mentioned, in the Hole and Warner Valley units, we don't see fire history being a significant predictor of bacterial community composition. Less than 10% of the permanova iterations returned a p-value of less than 0.05. And so in these two units, potentially there is sufficient time during those fire-free intervals for the communities to return to their pre-fire state. And so you're just seeing an effect of the most recent fire, and not an effect of past fires in these units. And like I had mentioned before, bacterial communities tend to be quicker to recover compared to other biotic communities, like soil fungal communities and plant communities, in part due to their overall quicker turnover times, where they can reproduce on the order of minutes, whereas it takes years for plants to reach maturity and reproduce. And then another potential driving mechanism for this is that the previous fires may have been low enough in intensity or low enough in burn severity that they didn't cause changes to soil properties that lasted, or changes to the above ground plant communities that were reflected in the two different treatment types. The two previous studies I'd mentioned that have looked at fire frequency and its effect on soil bacterial communities have found a correlation between differences in community composition of the bacterial communities and differences in the above ground community composition, and also differences in soil properties. And here we explicitly targeted plots based off of matching the dominant vegetation types. So we might just be like missing that essentially. Okay, so we looked at the communities as a whole, and now we want to look at the responses of individual bacterial taxa to fire. And we used a package in R called corncob, which uses a beta binomial regression to identify individual OTUs, which are operational taxonomic units, is like akin to a bacterial species. But basically, we are using this beta binomial regression to identify OTUs that are differentially abundant with the different burn severity and fire histories. And so before I show you guys the results, it's going to be a lot. So I am going to walk you through how to read it first. That vertical line is showing no change in relative abundance based off of fire history. And if a point appears on the right side of that vertical line, that means that that OTU was significantly enriched in the more fire plots. Whereas if it appears on the left side of that line, then it means that that OTU was significantly enriched in the less fire plots. The points I'm showing, all of them had a significant effect, and they will be colored by the phylum that they belong to, and then the size is relative to the relative abundance in the more fire plots. So we looked at this at the Butte Lake unit, and then also at the Hole and Warner Valley units. And in total, we identified 543 OTUs that were differentially abundant with fire history, and 80% of those OTUs were identified in the Butte Lake unit. You don't need to, nor should you be able to, read the y-axis, but I should have mentioned that they're grouped by family. But the key thing that you should take away from this is that the response of individual species of bacteria is not conserved at the family or the phylum level. So you're not seeing, there are some exceptions, but generally you're not seeing that OTUs from a phylum or from a family are always going to be enriched in more fire or always going to be enriched in less fire plots. We then looked at this with burn severity. Again, I'm withholding the data first to describe how to read this graph because it's going to be a lot again. But here we compared the relative abundance of each OTU compared to the lowest burn severity class at that unit. So in the Butte Lake and Hole units, that's going to be compared to the unburned samples. And in the Warner Valley unit, again, because we didn't collect any samples that were unburned, the comparisons are to the low burn severity. If the point is above that dashed line, it means that the OTU was enriched with that burn severity class. And if it's below that dashed line, it means that it was depleted with that burn severity class. So in total, we identified 565 OTUs that were differentially abundant in at least one burn severity class. and 18 of these OTUs were identified across all 3 of the units. Again, we're not seeing a consistent response at the phylum level. However, generally, if an OTU was enriched or depleted in one burn severity class, it had generally a consistent response with increasing burn severity. So you're not necessarily seeing lines that are kind of oscillating around that horizontal line. Okay, and so that's a lot to look at all at once and not particularly informative. So what we wanted to do next was look at 2 genera of bacterial taxa that had previously been identified as fire responders and look at their responses with increasing burn severity. So the first that we looked at were 2 OTUs from the Massilia genus, and Massilia has generally been identified as occupying a large amount of space within post-fire soils, just being dominant in post-fire soils across diverse ecosystems, including chaparral, mixed conifer forests, again in California, and then the Canadian boreal forests. And what we found is that 2 of these OTUs were enriched with increasing burn severity in 2 of the units. And another thing to mention is that OTUs from this genus have also been identified previously in fire literature as fast growers and being enriched with the addition of pyrogenic organic matter to soils. Next, we're going to look at Nova herbispirillum and 2 OTUs from that genus. And Nova herbispirillum, again, has been identified as a positive fire responder, specifically with its ability of OTUs within this genus to degrade pyrogenic organic matter. And this genus has been identified as one of 3 genera that have been consistently enriched with the addition of PyOM in multiple soil types. And some OTUs from this genus were identified to take up and incorporate PyOM into their biomass. In another study, they found this genus to be a late successional genus, essentially meaning that it increased in its relative abundance across time, from 1 to 20% in relative abundance within 9 months post-fire. And with our study, we found that it's enriched with increasing burn severity. At least one of those OTUs is enriched with increasing burn severity. And another OTU, OTU 210, is only enriched in the low burn severity class. So to kind of summarize the findings from my research, first, we found that soil pH increased with burn severity and was higher in the less fire plots. Next, we found that it was consistently a significant, burn severity was consistently a significant and strong predictor of bacterial community composition only in one of our units. So our hypothesis was supported in one of our units. We also found, again, that fire history was a significant predictor of community composition only in one of our units. But when we accounted for burn severity, it was no longer significant. And then finally, we identified bacterial taxa that were enriched in more fire plots and with increasing burn severity that were previously identified as fire responders. Okay, so some more takeaways from this research. Essentially, the effect of soil pH increasing with burn severity may in fact fade over time, but we did find that it did increase 1 year post-fire. We found that smaller spatial scales of burn severity were better predictors of differences in bacterial community composition. But overall, burn severity was an inconsistent predictor of community composition. And that one previous unshared fire between areas that were otherwise matched in their edaphic properties and their dominant vegetation may not have been sufficient to drive differences in soil pH and bacterial community composition. That's detectable 1 year post-fire at this time point. That's not to say that as the recovery trajectories continue, there may be, in fact, differences. But at this time point, 1 year post-fire, we didn't identify differences based off of fire history. Again, we identified taxa that were enriched with more fire and increasing burn severity. And we found that there was a weak effect of fire history on bacterial communities and soil pH, which may indicate that bacterial communities are in fact resilient to the contemporary interval of burning. So essentially, the fire recurrence within these areas may be sufficient to allow for the bacterial communities to return to their pre-fire state. Some future directions for this work. As Thea mentioned, we went out this past summer to collect samples again, so 2 years post-fire, and it would be interesting to see how the communities change across these burn severities across time. We're only looking at the bacterial community composition, but it would be interesting to look at the fungal communities and to link that to conifer recruitment in these soils. So just some future directions. And lastly, I want to acknowledge all of the people who have made this work possible, because it was not possible all by myself. Firstly, my advisor, Thea Whitman, has been fantastic and a great role model as a scientist and general human being. Thank you very much for your help with all of this. My committee members, Dr. Jingyi Huang and Dr. Monica Turner, for their feedback on the proposal and the progress of this work. Everyone in the Whitman lab has been super instrumental in the direction of this work and providing advice at multiple steps throughout the progress. Along with all of the soil science department and graduate students have been super supportive and have helped foster a newfound love of soils within my heart. So thank you. I also wouldn't have gotten this work done without the 2 amazing undergrad research assistants that I had the pleasure of working with for the past year, Sergio Luis Aviles and Lindsay Blanchfield, and all of the staff at Lassen National Park, Emma McClure, Dan Osman, Mike Magnuson, Keith Smith, Chrissy Frenzen. I also forgot to mention Varya Fedorova, for all of their help with the logistics of doing the field work and providing greater insight to the park itself and the histories of fire and how this ecosystem works. The UW Statistical Consulting Group, small plug, if you haven't yet used their services, you totally should, and ask for Nick, he's great. And finally, my friends and family for supporting me through this last 2 years and making all of this grad school experience possible and enjoyable. So with that, I have a thank you slide, but it has too many pictures to appear quickly, so questions. Yeah. Yeah, Doug.
Thanks, Isabella, for a great talk. I was really surprised to see the range of pH that you found from your sample sites. And I know that pH does have effect generally on microbial diversity. Did you investigate that directly, the correlation between soil pH and community?
Yes, that's a great question. So for people on Zoom, Doug was asking whether I investigated the effect of soil pH on bacterial community composition, because he was interested in the wide range of pH that was found across the sites. So to answer your question, yes, we did look at whether soil pH was a significant predictor of bacterial composition, and it was consistently across the mineral horizons for all 3 of the units. So it, again, in this data, we're also showing that it is the strongest predictor for differences in community composition. Yeah. I had another thought, but I'm forgetting what I wanted to say. So I hope that answered your question. Okay. Oh, sorry, another thing I mentioned. So the pH in the whole unit was significantly different and lower than in the Butte Lake unit. And so we are seeing slightly different pHs based off of the region of the soil that was collected. Alfred.
Thank you. So when it comes to the variance of the bacterial community, you think you explained it with the factors of sampling? You know, you sample by burn severity, by location, by depth of horizon. There was another one, the number of burns. You think those are good factors to explain it, given the relatively low percentages of the variance that you could explain?
I would.
First question.
Oh, okay. Should I answer that, or you want to give me the second? Oh, okay. So the question was, do I think that these factors that I described are good predictors for bacterial?
No.
They explain the variance. I would say the diversity of bacterial communities, and in previous studies that have looked at factors that explain differences in community composition, often they're reporting R-squared values that are much lower than what I was looking at for at least burn severity. And so I would say that like part of that might just be due to the depth of sampling that I was doing, where I was really characterizing the communities at like a fine, like more deeply than other studies have. But to your point, like 26% of the variance explained by a single factor. For like macroorganisms or like larger organisms, may not be a large variance that's explained by a single factor, but within a microbial community context, I would say that it is, it's fair. Yeah, it is a fairly good predictor, or like explains the variance well. Yeah, and part of that is just like the immense amount of diversity within these soil communities, and that we're sampling potentially not deeply enough with next generation sequencing to like fully identify the entirety of the communities. So there are just like limitations within microbial studies, but I would say that these factors are explaining the variance better than other studies that I've seen.
So maybe my second question is that what are the factors that you wish you had measured that would have given you a better explanation of the variance in that community?
Yeah, that's a great question. So what factors do I wish I had measured? I didn't mention it, but we were also looking at the total amount of carbon and the total amount of nitrogen in these soils. And I would imagine that these factors would also explain a good amount of variance, because they're more descriptive of the amount of like nutrients available to the microbial communities. So those two factors could be good explanations for differences in community composition.
Another suggestion, another question is maybe in response to that question too. If you have more intense fires, you get a coarser texture. If you have a coarser texture, you have a lower buffering capacity. If you have a lower buffering capacity, the effective pH lasts shorter, if you like. So I think the texture would be probably the easiest one to investigate and see whether that has any effect. The buffering capacity might be another one easily to investigate. If you consider the pH to be an important factor in explaining the community diversity. That was
Great. I love that suggestion. Thank you. Okay, Kelsey.
Soils. Yes, fire plots.
Yeah, so that's an interesting question. And I think that's for Park Service, fire ecology, it's very objective motivated. And so I think that including soil samples you'd need to think hard about like what questions you're asking before collecting those samples. And if you're concerned about erosion capacity, potentially like then doing some field-based tests would be interesting to incorporate. But maybe this isn't, I guess I'm a bit skeptical. If you're collecting samples with the purpose of characterizing the like microbial communities, I would have a difficulty understanding how useful that is at like a system-wide Park Service fire effects program level. But I do think that we should be characterizing these soils a little bit more than what's currently being done, which is quite minimal. So yeah. I liked the Butte Lake unit. I think it had great campground, beautiful cinder cone, nice hike. So yeah, I love that place.
We're cracking questions. We're at the top of the hour, so let's thank Isabella again.
Thank you. Thanks.